My name is Atique Ahmed. I'm an associate professor at Northwestern University, Chicago, and we've been working with this idea of chemoresistance for a long time in GBM. I think Michael did excellent review on GBM, so I'm not going to go into that. But these are the people done this work, especially incredible undergraduate Ella and Jack, who moved on already from the lab in a greener pasture, but they did a tremendous job of driving some of this project and data I'm going to share with you, and of course the funding agency. So as I said, Michael did a good job, so I'm not going to go through this. It's a very deadly disease. One thing that I want to mention is that the origin of these disease, yes, is still a controversial topic, that where this tumor coming from. And it's considered that they're coming from astrocytes, but at least in the animal model, when you transgenic animal model, when you induce oncogenic activations in our tumor suppressor inactivations, you can see both neurostem cell and astrocyte can give rise to this tumor. So it's still, we are not sure where it's coming from, but one of the things I want to mention is that probably neurostem cell is driving this gliomagenesis more pronouncedly than the astrocyte, but astrocyte also can do that. So still, it's a controversial topic, and we are still trying to figure this out, what's going on. So in the nutshell, if you think that why GBM so difficult to treat, I think Michael mentioned this, but I want to re-emphasize this this issue, that why GBM is so difficult to treat. So one of the things is blood-brain barrier. Not every chemotherapy can cross the blood-brain barrier, so we have very limited arsenal against this disease. Second thing is immune microenvironment. Is a very immunosuppressive microenvironment. And some of the PD-L1, PD-1 checkpoint blockade that work in other cancer is not working for GBM. It is a huge topic, and tremendous effort has been invested on this idea of how to activate this immune microenvironment. Second thing, as previous speaker mentioned, is that it's incredibly heterogeneous. Not each cells are equal in the same patients. So what happened that they don't respond to therapy equally. So you end up having very difficult problem where that you kill some population, but other cells stay alive and then give rise to this recurrence. And finally, therapeutic resistance. These cells are incredibly adaptable, and I'm going to talk about a mechanism we think that's playing a role in this adaptability. So chemotherapy, we work in, we use this as a model, temozolomide, because this is the standard of care for GBM. The Roger Stupp, who was in Switzerland, University of Chicago, Switzerland, now in our institute, who kind of established this temozolomide as a standard of care. So temozolomide damaged this DNA by creating this adduct and these three adduct. You can see the percent of adduct they create. But what is interesting thing I want to point out is that this 5% adduct that create 06mg drive this effect of temozolomide, anti-tumor effect of temozolomide. And cells find a way to remove this adduct, MGMT, one of the enzyme that's been produced by the cells that can remove this adduct. And this serve as a biomarker where MGMT methylated promoter, where they cannot express the MGMT, patient can survive longer. Okay, MGMT unmethylated one the patient's GBM progress very aggressively. But what is interesting thing is that regardless of this MGMT status, patients eventually circumvent with this disease. So saying that there's alternative mechanism of resistance, such as Michael showed, and I'm going to show you another mechanism of resistance. So the idea of this glioma is a two model that has been proposed. One is that this stochastic model, where it consider as each cells within the tumor are equal and they give rise to tumor. Okay. However, there's a recent model that been gained a lot of attentions, which is the cancer stem cell model. This is a hierarchical model where it's considered that within the tumor, there is a stem-like cells that is the main culprit and considered to be driver of this gliomagenesis. And it has been shown that this glioma stem cell has a self-renewing capacity, high DNA repair capacity, and also express high level of efflux pump in their surface, so they can pump out this chemotherapy. And the non-glioma stem-like cells shown here is I don't know. Can you see my arrow? Yes. Maybe I can put it.
Yes, we can pointer.
See. Okay. So a non-glioma stem cell, which is rapidly dividing cells, can be targeted by chemo and radiation. So you get rid of this. These cells stay silent, so they can again give rise to these differentiated, highly proliferative cells and initiate recurrence. So now, one of the things that I observed when I was a postdoc, this is my postdoc project. I did it over, I had another project that I'm doing on from PI, but this was my observation, and what we were able to show is that observed that under chemotherapy or temozolomide stress, the cells start to become stem-like cells. Okay, so this is a neurostem cell culture in the neurobasal media. And if you put them in without neurobasal media in the temozolomide, they start to form like ball after 6 to 8 days later. So one of the questions I was wondering is that how these cells behaving like stem-like cell. So we generate this reporter system where you have RFP gene driven by this glioma specific promoter, and we express this, and we stably transduce these cells, and you can see that these ball-like cells or neurosphere-like cells under temozolomide express this gene, activate this gene promoter that drive this glioma stem cell niche. So one of the questions that I ask is that whether these cells are converting or expanding within the glioma stem cell populations. So this is a very controversial topic of marker of glioma stem cells because it is very, very plastic. These cells are extremely plastic, so they change the marker. One of the marker that we are using for marking these glioma stem cell is combination of CD133 and CD15. Both of them are surface marker. And if you take these negative cells that don't express this glioma stem cell, we ask the question that what happened when you take these negative cells and expose this temozolomide. And you can see that we sort the cells out, and the double negative cells, you can see day 2 post sorting, they're very, very low. And the yellow bar is a control, and black bar is exposed to temozolomide, physiological dose, which considered to be about 50 micromolar. And what you see is that after 8 days, there's a huge expansion of these double positive cells, yes? But what is, to our surprise, what was really surprising, this double negative cell becoming the not double positive cell. So that is the core topic that I'm going to discuss going forward. What we're showing here is that there's a two possible way of expanding glioma stem cells. Yes, one is selection, where you deplete this rapidly dividing cell and expanding this quiet quiescent cell. Second thing is that expansions, where you selectively expand these glioma stem cell populations. But our study throughout last 8 years proposed a new possible mechanism where there is a non-glioma stem cell, rapidly dividing cell under therapeutic stress, can expand the population of glioma stem cell. So this is now, when we try to propose this idea, there's a huge push against it because everybody thought that there's a static populations of glioma stem cell, and they liked the idea because they thought that they could be druggable. But we have shown over and over again that these cells expressing this reporter system come in and out of this glioma stem cell state. And we can track this in vivo in the animal model, and you can see that these red cells, this is a SOX2 promoter, red cells staying very close to, perivascular niche, close to the vasculature. That has been proposed, that in vivo niche for this glioma stem cell. And what is interesting is that these, when you sort them RFP positive cells, you can see that they are expressing, or they can form tumor when you engraft them very, very small amount. Here we are implanting 100 cells, and 9 out of 7 animal develop tumor, whereas CD130 positive cell, 100% cell express CD133, also 3 out of 3. So they're saying that this RFP positive cell do possess this tumor initiating capacity. And what is the most important thing that I want to show you, this is probably, how can you prove that in clinically that this is relevant? Yes, we are still trying to do that, but it is very difficult question to do clinically. But at least in animal model, what we show is that if you put a suicide gene driven by this promoter and you deplete this during therapy, what you see is that this is a TMZ plus glycocycovera, where you're depleting these cells during TMZ therapy, and you expanding the animal survival as compared to just TMZ. So what telling you is that this is a mechanism that may be possibly driving this recurrence and resistance. Now, why this is important, okay? So as Darwin say, it is not the strongest of the species will survive, it is the most adaptable. So what we believe that this plasticity, where glioma stem cell in and out of the state of different conditions can drive this adaptability and chemoresistance. I think that's why we think that this is a very, very important topic to target this plasticity, because this plasticity make these cells a moving target, which will be very difficult to hit with any therapy. So last 8 years, we try to understand the mechanism, how this is happening. And we really believe that it's a transient mechanism. Let me see, I'm on time. I have probably 2-3 minutes left. Let me see, I can show you some data of mechanism, how is things happening. We really believe that this is epigenetic driven mechanism, okay? What happened is that epigenetic barrier from transition from glioma stem cell state to differentiated state is very high. But during therapy, somehow this barrier is lowered, so they can transit between this state very, very quickly. And we have shown that PRC2 complex EGS2, which deposit the K27 trimethylation, open and closed chromatin, driving these adaptations, okay? When you block this EGS2 activity by drug, you can block the expansion of this glioma stem cell-like cells. And it has an in vivo therapeutic efficacy, but what is the problem is that, at least in our hand, none of this EGS2 PRC2 complex can penetrate the blood-brain barrier. So blood-brain barrier issue, I don't think this can be translatable. So now the question is, we asked that what is EGS2 PRC complex is controlling that, giving this plasticity and therapeutic resistance. And we did a simple experiment, knocking down EGS2 and hit them with temozolomide. Ask the question is, what gene is regulated? And one of the top gene came out is this ARL13B, which is a ciliary protein that regulated during TMZ therapy. So ciliary protein ciliary is a sensory organelle that expressed by many cells, and it is responsible for very key signaling pathway like sonic hedgehog. And ARL13B is stabilize these structures and also responsible for cargo movement. It never been investigated in GBM, and what we can see is that during TMZ therapy, the ciliary structure is elongated. Not only that, if you knock down ARL13B, you can significantly prolong the survival of animal and block the glioma growth. This is very interesting because at least in vitro, these cells grow absolutely fine, no problem at all. But in vivo, they cannot engraft anymore. So now we have shown that the EGH2 epigenetically binding this and regulating this ARL13B protein. I'm not going to go into detail, but the question is, what is ciliary protein doing it? So we did a mass spec analysis, pulled down ARL13B and did the mass spec during TMZ therapy. And what we're able to show that it interacting with a very important protein called IMPDH2, which is a de novo purine biosynthesis rate limiting enzyme. IMPDH control this de novo purine biosynthesis, and purine is very important for metabolism of DNA and RNA. And to follow my talk is a very complex process. You have to know two things. Purine can be synthesized de novo from the scratch ribose 5-phosphate, or purine can be recycled from the high energy molecule like RNA, DNA, ATP. In brain, and the purine salvage is predominant because it requires less energy. So they recycle purine. Okay, so now the question is why these cells try to regulate this purine during TMZ therapy. And we showed that ARL13B is interacting with IMPDH activity, IMPDH2. Now question is why ARL13B interacting and what it is doing it, okay? So what we believe is that the purine recycling and purine salvaging is very important because as we know, the TMZ alkylate this purine. So now we could ask the question, what is ARL13B doing? So what we did is like we knocked down ARL13B and do the radio level tracing. And what we're able to show here is that, and then we track this radio level in the DNA and RNA. And what we're able to show that, and we all did all this thing, but quickly what we're able to show that, that if you knock down ARL13B, what happened is that this purine salvage is unleashed. So both RNA and DNA, and what we believe that what's happening is that ARL13B interact with IMPDs to block this purine recycling and force the cell to switch to de novo cycling during TMZ therapy. De novo, remember, they're making the fresh purine, okay? So why cell wants to do that? What we believe that during TMZ therapy, because this is purine is alkylated, if you recycle this purine, cell get damaged, okay? The DNA will get damaged. So cells somehow sense that. That's why they switch to purine de novo synthesis where they make the fresh purine. And we have shown that experimentally that this is exactly what's happening, is that this O6MZ get incorporated in the DNA if you remove ARL13B, and it's shown by mass spec. So the summary is that take-home message is that ARL13B is regulated by epigenetic modifications, and that leads to blocking of the salvage pathway, and that's unleashed this de novo pathway, thus protecting the cells from this using the damaged purine and DNA damage. And now question is, how can you translate this in animal in patients? So actually this pathway can be targeted by a immunosuppressive drug called mycophenolate moafetol that has been in the clinic already for transplant setting, okay, for kidney transplant. So now when you combine this with temozolomide, what we find is that these become more sensitive to TMZ. Okay, and we can completely protect this animal for developing tumor when you combine this MMF, mycophenolate mofetal, with TMZ. Based on this, we launched a clinical trial. Now we recruited almost all the patients. Now we're analyzing data. We're very excited about this. And then we'll publish the data pretty soon, but their data looks super promising. No side effect. Patient tolerate very well, and we are also receiving some nice signal that might justify you combining this MMF with TMZ for glioma therapy. So I think I'm gonna stop here. So summarize my talk is that I'm sorry I went a few minutes over, but interconversion between differentiated glioma cell to dedifferentiated cells as a mechanism of resistance, and we show this mechanism of resistance is driven by epigenetic plasticity, and that leads to this blocking of the purine salvaging and unleashing the de novo purine salvaging. And you can now block this this de novo pathway by MMF, which leads to sensitivity to TMZ. So thank you for your attention. I'm happy to ask you, give you and answer any questions you have. And I appreciate this opportunity for presenting our talk. Thank you.
Thank you very much, Atique. Very interesting speech. Maybe I start so the people, if they have a question, they can ask later. So you show us that the way of escaping of the glioblastoma cells is like the differentiation to the stem cells, that the mechanism behind this differentiation is like the changing in the metabolism of purine. And you show us also that you perform some trials with the drugs that blocks the de novo synthesis of purine, and together with telozolomide. So my question is, in the patients, you didn't have some effects like immunodepression, because all those drugs that are used for in the block of purine normally are used to downregulate the activity of the immune system. So you haven't noticed that in the patients?
So that's a very, yeah. So that's a very important, good question, because the question you're asking is that, okay, MMF is an immunosuppressive drug, and TMZ also we know is an immunosuppressive, yes, myelosuppressive. So when you combine them, you should have more severe immunosuppression and patient gets sick. Very interestingly, what we see is that with respect to immunosuppressions, the dominant factor is TMZ. So if you add top of MMF, they don't, the patient don't suffer more immunosuppression, which is supposed to, but actually the immunosuppression patient in this case is driven by TMZ. So adding MMF, we are now, I think the maximum tolerated dose, which is 2000 milligram per BID. I think this is the last cohort of patients. So far, no toxicity. Patients are doing
perfect.
Great. Yeah,
sounds really good, really promising.
Yes. So it is about we are at 25 patients. None of them show any dose limiting toxicity.
Okay, thank you very much.
So regarding that, one argument that people make, especially at Duke, is that immune suppression, that myelosuppression can actually be utilized in that when you've got that gap, when the big drop in leukocytes, there's a rebound effect. It's called homeostatic proliferation, whatever the hell that means. But the idea would be that you could then sort of vaccinate into that hole, and the cells that come out of it are actually more, they're less immune suppressed and they're more easily stimulated. So I'm wondering, have you tested any of this in an immune competent model to see if there was a way to sort of combine an immunotherapy with it? Because it seems
No,
like
yes.
immune suppression may be exactly what you're wanting to do, is suppressing the appropriate cells and allowing the cells you want to respond to come out of it.
Yeah, exactly. So yes, yes. So you're talking about the Duke idea of that. So what they do is like create space in the immune compartment by depleting the cells, pre-existing cells, and then expanding the re-educated immune cells in the compartment. No, we did not try, Michael, because that get really difficult to get approved by the FDA, because this is a first in patients trying. So we're not thinking that way yet. We want to see this, but that's a good idea. Yes, I think that's a logical step of combining this with the PD-L1 and see whether you can re-educate the immune system, because you're going to have a more compartment space in the patients.
Well, and if I can just dominate the conversation here and throw more questions out. So years ago, Heidi Phillips showed that when everybody was all about taking the stem cells and showing that the stem cells, when you put them intracranially into nude mice, it took 100 cells to turn into a tumor, and the non-stem cell, whatever they chose to call those wouldn't grow. But she actually showed that they would grow. It would take months for them to come out. And so I was kind of wondering about that dedifferentiation phenomenon that might actually, this seems to just sort of affirm that there is this kind of dedifferentiation that can go on. But regarding the fact that the going through the de novo curing pathway versus the salvage pathway, I think one thing we need to bear in mind is that due to lots of weird things that go on with the purinergic receptors on tumor cells, there's a lot of ATP in the extracellular environment. So tumor cells have almost a glut of ATP to absorb, and they'll eat this like candy, and it doesn't matter where it comes from. So I think that the de novo pathway is not a problem for the tumor to carry that out.
Exactly. So you're absolutely right that they have a lot of extra the metabolite in the microenvironment to feed them. Yes, and you show that they switch to lipid metabolism. Yes, even to produce the energy. And we find the same things when cell de-differentiate. Yes, into cancer stem-like cells, they also switch to lipid metabolism, and they prefer lipid metabolism over any other metabolism. So we still don't know why it is happening. But going back to your ATP, is that one of the things that we're struggling is that, this salvage pathway and this damaged nucleotide, how the cell sensing this? Yes, this purine is now alkylated during temozolomide. That's where they're switching from salvage to de novo. That's our hypothesis. But one thing we're struggling to understand, the sensor for this alkylated purine, that how the cell, it's kind of boggled my mind that cell can sense this simple change of this one alkylated purine, and then switch suddenly to de novo pathway. So I don't know. I don't have the answer for you. Yes, there are lots of ATP there, but how the cell sensing that, I don't know. I don't have the answer.
That's an excellent point. And I'll stop after I say congratulations for getting this moved into the clinic. That's just awesome. Yes,
it is. As a basic scientist, is probably most gratifying thing for me, seeing, because it took us almost 6 years, but it's in the clinic and it's almost done. So we are very excited about it, because at the end of the day, we get caught up with this idea of publication grant and this and that, and forget of the real purpose that we here is about for our patient. Yes, and as a basic, sometimes we kind of get distracted about this from all this noise.
Well said.
Yes, I think one more question.
Thank you, Ahmed, for your nice talk. Regarding your last proposition, how the cells sense and how they make decisions. From bioinformatic point of view, I think the cell communicate. They are always in the communication. So probably some network analysis and how they communicate by the groups, like at the single cell level, and how they communicate and what ligands and receptors are affected will help you to answer this question. But
No,
of
you're
course, you
absolutely.
need to have.
Yeah, you're absolutely right. I think and now we have the tools to study the single cell analysis. I think more than single cell analysis, I'm very excited about this spatial transcriptomics. Yes. Even though we are not getting the single cell resolution, but I think we should get it. And we are getting some exciting data to understand how the cells are communicating with this single cell spatial transcriptomic analysis. And we are getting some idea, but we're still not there. But you're absolutely right that how the cell communicate, they live as a community, and we still don't know their language. Unless we know their language and how they're communicating, I think it will be very difficult to treat this disease. And we are getting there, but I think you're right, absolutely.
Exactly, you are right about the spatial transcriptomics too, because this will give you the idea of the position of cells in space, where they are located precisely. And this is an advantage to do spatial transcriptomics and then to look at the communication between those cells and group of cells.
Yes, I agree with you 100%.
Thank you very much.
Thank you.